Thank you for the opportunity to present at this international forum. Today I'll be talking about challenges and opportunities related to newborn screening for Prader-Willi syndrome and other chromosome 15 imprinting disorders. Here are some of my disclosures. I'm an inventor on a number of patents related to newborn screening technologies. I've also acted as a paid clinical trials consultant for Bellberry Ltd and Optimogen Medical. And below I've included a number of grants that have been funding our research. So before I talk about newborn screening, I'd like to touch base about basics, underlying genetic causes of Prader-Willi, and standard of care diagnostic testing performed for Prader-Willi and other chromosome 15 imprinting disorders. So Prader-Willi is caused by changes in chemical changes at this locus 15q13 called methylation, which silences genes within this region. So in neurotypical individuals, methylation at this region is roughly 50%. Maternal one is methylated, paternal is unmethylated. In Prader-Willi, this region is hypermethylated, so methylation is approaching 100%. And this could either be caused by a deletion of a paternal copy, maternal uniparental disomy, or imprinting center defect, which is a methylation change without copy number changes on the paternal chromosome. Angelman syndrome is reverse. It is associated with complete lack of methylation at this very same locus, and it can be caused by either deletion of maternal copy within this region, paternal uniparental disomy, so 2 copies from the father, imprinting center defect, methylation, lack of methylation, maternal chromosome, or a mutation of this gene called UBE3A. Standard diagnostic testing uses DNA methylation of the SNRPN promoter located within this region to diagnose most individuals affected with Prader-Willi syndrome and roughly 80% of individuals with Angelman syndrome. And the techniques usually used are called MS-PCR and MS-MLPA. So if a child has symptoms that a clinician suspects to be either Prader-Willi or Angelman syndrome, they refer those individuals for this testing for molecular confirmation. Based on these referrals for testing, it has been estimated that the prevalence of each condition is roughly 1 in 15,000. Once methylation is confirmed, the DNA samples are sent for genomic testing called microarray, which defines etiology. It can also identify another condition called G15q syndrome, a common cause of autism. G15q syndrome is caused by duplication of this very same region that is either deleted or imprinted, abnormally methylated in Angelman's and Prader-Willi. Only 1 to 5% of individuals with Prader-Willi and Angelman syndrome have been accounted to methylation mosaicism, so imprinting defect. Methylation changes without copy number change. This is a likely underestimate because individuals with methylation mosaicism have atypical presentation, and some may not have been referred for appropriate testing. This is an issue that would be addressed through newborn screening. So now I'll talk about more general newborn screening workflow currently available. Virtually all infants born in Australia, Europe, and United States are screened for over 25 rare disorders, but not Prader-Willi syndrome, as part of state-sponsored newborn screening programs. The primary goal of these programs is to reduce mortality and morbidity due to selected rare disorders through early diagnosis and treatment in the first year of life. Stringent criteria have been developed that support selection of conditions to be included in these population-wide programs. With recent advances in genetic and genomic testing, these original criteria have been updated. Now, additional rare disorders can be identified using genetic genomic testing, for which early detection may benefit affected newborns and their families. SMA, or spinal muscular atrophy, is a great example. Recent development of a successful gene therapy for SMA has resulted in it being included in state-sponsored newborn screening panels. For Prader-Willi, there is a treatment already from birth, growth hormone, that makes a big difference and supports a strong case for Prader-Willi to be added to standard of care newborn screening. Gene therapies are now also being developed for Angelman syndrome, providing further justification as Angelman is identified by the same test as Prader-Willi. So why would you want to do newborn screening for Prader-Willi and other disorders? Well, the main reason is early age of diagnosis is usually equal to leads to early intervention if available. Growth hormone in the case of Prader-Willi, that leads to improved outcomes. Another reason is that unbiased screening of the population independent of social economic factors, and this means equity. So regardless of where you are located, developed or developing country, whether you live in rural or country settings, and regardless of the access of medical support that you have, you will be identified through newborn screening. This may not be the case through standard diagnostic testing. Because if you're not identified as likely to have Prader-Willi syndrome or Angelman syndrome, you will not be referred for appropriate testing. Another reason is accurate prevalence estimates. So if you have better estimates than what we have right now, reflective of what's in the general population, it may justify improved support. Another reason is prevention of diagnostic odyssey. So this would lead to less anxiety and stress for the families if they receive diagnosis early. Advice to the families about future reproductive risks is also quite relevant. Not so much for Prader-Willi, but it is for G15Q, another condition that would be picked up if you screen for Prader-Willi, and these benefits has clear benefits for future generations. So what are the critical gaps to make this happen? While newborn screening makes sense for many rare disorders, state-sponsored newborn screening programs are unprepared for this, especially if many conditions, including Prader-Willi, are identified. This is particularly the case for genomic testing. So critical gaps in knowledge are lack of a low-cost accurate screening test that is scalable and that is informed by optimized population, clinically meaningful cut points. So you only identify individuals that benefit or more likely to benefit from this, and not the ones that would not benefit from earlier diagnosis. Lacking knowledge of natural history at population level. This would include mild or subclinical cases less likely to experience the benefits of screening, but equally exposed to all its harms. Another area that is lacking in evidence is whether benefits of early newborn screening outweigh its harms, including costs. So now I'll sort of discuss briefly technical requirements for newborn screening and costs. An important limitation of newborn screening at present is availability of low-cost test that's around $1 per condition per infant that works on as little as a single 3mm newborn blood spot punch. Usually 5 blood spots are collected per infant, and each one has about 10 punches. After the standard of care screening for 25 conditions is completed, there isn't much material left. So it needs to work on a single 3mm punch, which is what you can see here. So together with the cost, this largely precludes standard diagnostic genomic testing, including chromosomal microarray, whole genome sequencing, and whole exome sequencing. These techniques require quality, non-fragmented DNA extracted from usually 10 or so newborn screening punches. To be able to extract DNA, it's usually done using commercial kits, which in themselves cost roughly $10 per infant per extraction. And on top of that, you have the cost of the test, which is roughly It ranges between $20 and potentially up to thousands of dollars for whole genome sequencing. And finally, quantity of the available material is an issue and would be insufficient for most of these tests, as you would generally have only 1 or 2 3 millimeter punches left for additional testing. Off-target effects, ethics, and follow-up costs are also important gaps that need to be addressed to make newborn screening for Prader-Willi happen. For genomics, identification of genetic changes that cause other unrelated diseases is a significant issue if genomics is used as first-tier test. Identification of genetic changes of uncertain clinical significance is another caveat. And this is because if you have such changes, you need to follow them up using additional laboratory, clinical, and bioinformatics pipelines that cost significantly, that add significant costs. If these genetic changes are related to late onset adult conditions of incomplete penetrance, disclosure could be harmful to the infants and the families. Another issue with genomic testing is the information. If you use it as a first-tier test, you will have information on things like height, potentially whether your child will become bald when they grow up, Alzheimer's disease, cancer risk, stroke, lots of different things that are not related to the condition screen. So if you use genomics as a first-tier test, you will need to have extensive pre-test counseling for all parents. You will also need to have extensive clinical follow-up for first-tier positive cases. Together, this will add significant cost and complexity that is not compatible with current newborn screening programs. Moreover, a significant number of infants with Prader-Willi due to UPD and imprinting center defect, so the methylation changes without copy number changes, will not be picked up by genomic testing unless these are coupled to methylation analysis. An alternative solution may be a hybrid approach between targeted first-tier methylation testing followed by genomic confirmation to confirm etiology. These two papers, recently published papers, explored use of MSMLPA and high-resolution MELT analysis on DNA extracted from blood spots for newborn screening for first-tier testing to detect Prader-Willi and Angelman syndrome. They're primarily focused on showing what is the best method to extract DNA from blood spots to do methylation analysis. This particular paper is where we actually developed our own. This is from my lab, where we developed our own technique called MSQMA that does not require DNA extraction and was shown it was feasible on roughly 17,000 infants from the general population. So this particular approach was a hybrid approach that I've talked about. First-tier methylation testing, followed by genomic testing for confirmatory analysis. Advantage of this was that we were not picking up conditions that we were not looking for, and primarily only identifying conditions that we were screening for that would benefit from earlier diagnosis. So in conclusion, broad genomic testing that will identify some infants with Prader-Willi does not fit current screening workflow requirements. If genomic testing were to be introduced as a first-tier test, new system will need to be established to address these issues, including newborn screening material requirements, costs, ethics, consent, and follow-up. Targeted first-tier testing using methylation analysis for Prader-Willi and other conditions justified to be included in newborn screening may be better fit for newborn screening requirements and systems already in place. Future population scale research is needed to perform cost-benefit harm comparisons between these models and the current diagnostic standard of care testing to determine what is needed to bring newborn screening for Prader-Willi into practice.